So in today's symposium, we could see a wide range of topics. So starting with basal ganglia, followed by uh, mm, uh, spinal cord based muscle, and then you had hippocampus, and there was also databases. The last presentation being more of cerebellum, and I'm also taking the similar path with cerebellum, but my question was a bit different. So with the recent advancements in neurorobotics, especially in the case of neuroprocesses, where human physiology as well as behavior is considered together, how can we control a low cost neuroprosthetic device? So that was my key question, and for which I used a cerebellum inspired model. So before I jump into all these concepts, the first thing to identify or understand why a model is important. So in the case of computational neuroscience, you know that there are not only in the case of neuroscience, any other scientific areas, you have enormous amount of data in all forms, may it be the structure, the function, the spiking behavior, the circuit level. Any of these things account to the data, which uh, all these data will join together or give some kind of information to us, and what it is conveying to us can be understood only with the help of a model. And uh, from like 1960s onwards, there have been a number of models that have come up based on the cerebellum, starting from James Albus's CMAC, cerebellum-inspired motor control theory. And from there to an adaptive filter in the 1982 by Fujita, and so forth. So there were many models that were developed. So based on these data and model, people have come up with different theories, brain theories. So starting from 1906 with the reflex action theory, which is the simplest as well as the widely known theory. And again, you have a lot of other theories. So the main theory which we would be focusing is motor adaptation, or motor programming, or motor learning theory. Okay. So before we go into that, there are two, actually, the modeling approaches differ in different platforms. So, like Professor Naldi was saying, more of a top down approach. I usually look at a bottom up approach, where I would be collecting more of data and coming up with a realistic model like a Hodgkin-Huxley or a point neuron kind of model, which can be introduced to a robotic model and then come up with a hypothesis which would later lead to a theoretical model. Okay, so this is a general idea about how the modeling approach happens. So the question here was motor control, and like so in the morning class, like when Srinivas Chakravarti was saying, the cortical loops take more time to process the information. The similar kind of information was put forward by Kawata also in the 1992, when he was studying on VOR. So there he referred as the cortical loops take around 300 milliseconds more when compared to the cerebellar circuits. So uh, the cerebellar circuits are known for its fine tuning, and the cortical circuits are for the initiation or the motor planning kind of behavior. So the during the learning process, so in the case of motor control, there are a lot of areas that are being involved. So we have covered around cerebellum, basal ganglia, spinal cord, and muscles already in the set of previous talks. The only thing left is the cortex, which is again a very vast area. So whatever we perceive through our eyes are being taken to the visual cortex. From there, it is being projected to the motor cortex, and so forth. So this loop is being what is studied from different by different researchers. And if you have looked at Daniel Wolpert's TED talk, he had specified that the main use or the only use of brain is actually movement. So whatever you perceive or whatever you do in life, it ultimately leads to some kind of movement. It can be a muscle movement or some kind of movement related activity. So it is very important to look at the motor control aspect of it also. So during this process, after the learning is complete, you say you can do a task without any error. So in that case, you say it as a motor performance that is being carried out. So example is a person walking through a rope, or might be you are riding a bicycle. So all these are fine tuned works or aspects. So when you are not learned completely, it tends to come up with errors, and you call it as motor errors. And during the motor learning process, you have three different stages that happens. So this was put forward by Fitz and Posner in 1967. So during a learning process, you have the first or the pre-training phase called the cognitive learning, which where the stimulus is being presented and the output is being predicted, and it can be erroneous also in nature. In the second phase, that is in the training phase, it is called as the associative learning. So in the associative learning, the errors are being corrected. And the last phase, that is the post training phase, is called the autonomous learning, and that is when you say that this model is trained. And based on this, uh, people have, or researchers have come up with different learning theories or machine learning algorithms. So one of which is the supervised learning algorithm. Mhm. Okay. Supervised learning algorithm, which was a learning algorithm that was only for the cerebellum, or was kept only for the cerebellum, but things have changed. I'll tell you in a couple of slides. And in the second type of learning was the unsupervised learning, where uh, you have similar kind of patterns. You join all those patterns together to come up with some kind of understanding. So that is the unsupervised learning, which was confined to the cortical regions. And the last one is the reinforcement learning, where reward based learning or a punishment based, critic based learning happens. So that is what we had seen in the basal ganglia. So this was the general kind of learnings that were uh, uh, put forward in the beginning itself. Okay, so we were interested in a cerebellum inspired learning because we were looking at a motor control. When a robotic arm or a neuroprosthetic device moves that side or this side, it is mainly for the fine tuning that matters. So we were interested in looking at the cerebellar microcircuitry, which is a well structured circuitry, which is very modular in nature, and it is more like a repeating unit that is seen. So more like a processor kind of a system. So you have input, two types of input that is coming into the system, one through the mossy fibers and the other through the climbing fibers. So the mossy fibers carry in the sensory signals, and these signals are being recoded in the granule cells, which impinges on the Purkinje cells, which is the sole output of the cerebellar cortex, and from there to the DCN, which is the output of the cerebellum. And the errors that are formed during this process is calculated and sent back to the Purkinje cell via the climbing fibers, that is the extensions of the inferior olive. So, and this substructure was very important from 1960s onwards, when looking at the uh, discoveries of John Eccles in the 1950s, David Marr proposed the codon theory. So that was a computational insight that was put forward. And he said that this is error-based learning that is happening, and which was proved by James Albus in 1970s. And he said that this is more of a supervised learning. And later in 1982, Ito came up with the experimental evidence for that. And he specified the uh, synaptic plasticity, that is the LTD and LTP. LTP was found earlier, and LTD was found by Ito. And these three parts, or these three researchers together, put forward the Marr-Albus-Ito theory. So that is the theory which has been running from past 50 years or so. So after which we have come up with different discoveries also, but we have not like uh, we have not attempted to change or modify these kind of Marr-Albus-Ito theories. So in general, cerebellar function can be considered as a predictive controller, which could generate an internal model of the body dynamics. So you can create a internal model out of it, and based on the sensory feedback, it can learn and refine the motor commands as well as adapt the synaptic junctions. So uh, the uh, the neuron numbers are also to matter. So there in Ma's paper, Mar 1969 paper, he said that it around 250 million mossy fibers give sensory input to the around 50 billion granule cells, which is very huge. That is why he said it as expansion based on recoding. And from there, it is from this 50 billion granule cells, it is being impinged onto the Purkinje cell. And from the Purkinje cell, it is being connected to the DCN, that is the output. And the output is very small in number, that is 1 million DCN. So during this process, there is something called as input output association that is being stored, and that is happening in the Purkinje cell. So, in the cerebellum, there is an provision to take up multimodal input. So, may it be in visual, auditory, somatosensory, or even efferent signals. Whatever kind of input, sensory signals that comes in, passes through the cerebellum and has a role in motor control. And this happens through majorly through two pathways, that is the corticopontocerebellar pathway as well as the cerebello-thalamo-cortical pathway. So these are the CPC and the CTC pathway. So which is the pathway that comes into the cerebellum as well as leaves out of the cerebellum also. And from here, people started looking at how is that the cerebellum is performing this fine tuning movement. So from there, people came up with the control schema. So that was a breakthrough, that is in 2005, where they suggested that from the higher centers of the brain, when information comes in, the motor planning is initiated, and from there the coordinates are being undergone with transformations to give the motor commands. So these motor commands are being sent to the cerebellum, and from there the motor control happens. And based on the error or the changes that has happened, it is sent back to the through the loops to correct the particular system. And like I told you earlier, there have been different models that has been associated, starting with the adaptive filter model, so which suggested that Golgi cell played a major role in the timing of timing of the movement. That was the adaptive filter model. Then Cavato in 1992 put forward the multi-forward inverse model. So here they introduced an internal model and a forward model as well as an inverse model, which would work in pairs. So our body also works like that. We have multiple pairs of forward as well as inverse model, which will work together to attain a particular motor task. And later, then we have many other models. So one of the recent model was not the very recent, was Pedrocchi 2006. So that is called the error mapping controller. Followed by you have Edlet and Spinnaker. These are all different kinds of models that were developed. So we also started looking at something of this sort. We wanted the a deep learning kind of working, as well as the cerebellum inspired part, to come up with doing a motor control task for a low cost robotic arm. Okay. So the first thing we had to do is to come up with the reconstruction of a neuron. So in the case of neurons, we have different kinds of models that are available. It starts with the non-linear cascade of neurons to a compartmental model to a single point neuron model. So Hodgkin-Huxley model has been widely used. So in a mostly the platform they have used is neuron, and there have been a series of 4 different equations to come up with the firing behavior of that particular neuron. So in the case of point neuron, this is far reduced to two differential equation, one for an adaptation, one for the voltage calculation. So we looked at both Hodgkin-Huxley as well as the ADAX model. So adaptive exponential integrate and fire was the model which we used. And the first step was to identify or come up with the parameters for the ADAX neuron. So we reconstructed, or we came up with the parameters of most of the neurons in the cerebellum, most of the important neurons in the cerebellum, that is the granule cell, Golgi cell, Purkinje cell, the inferior olive, and the DCL. With a set of coupled equations as well as a reset function, and optimizer that was used was a PSO algorithm. That is particle swarm optimization algorithm, and the fitness function or the condition was a coincidence detector was used to identify whether we have attained the correct firing behavior or not, based on the interspike interval as well as the timing and the shape. So this is how, and we validated it with the help of a F versus I graph, as well as the first spike latency. So the first one is the granule cell, and so these are the different cells. And then we also uh, could capture the granule cell firing behavior. So there were experimental traces that were available with 4 excitatory as well as inhibitory connections, the different range, series of inhibitory connections, and the firing behavior was replicated as such. So that was the first step. And followed by that, the next thing was to convert a biological scenario to a mathematical abstraction. So we had this circuit, the cerebellar circuit, with the mossy fibers giving connections to the granule cell and to the Purkinje cell. And so the circuitry was well explained earlier. And based on that, we came up with a design, and we named it as DL-cis-NN, Deep Learning Cerebellum Inspired Spiking Neural Network. So here the input was encoded. The first step in the circuit was to encode the input into a convolution layer, which mapped onto a granular dense layer. And then the information was pulled up in the Purkinje layer. The output that was obtained in the Purkinje layer was decoded back with the help of a decoder, and the errors were calculated. So the first step was encoding, and we use different sets of encoding schemes, starting with the BSA. BSA algorithm is Ben Spike's algorithm. So there, every feature was converted to a particular spike, and then this was introduced. Whereas in the case of granular layer encoding, it is more like a lookup table. So that was again put forward by looking at the different firing behavior of granule cell, and we developed the spiking behavior based on that. The third kind of method was the granule, the Gaussian kernel based encoding. So the Gaussian kernel is taking a feature and making different neurons out of it. So more like you have a normal distribution kind of behavior for the firing of the neurons. And recently, we have also looked at one hot encoding scheme that was to come up with the visual as well as auditory stimuli. So that was shown in Ishikawa 2015, and we could also reconstruct the same using one hot encoding scheme. We also looked at the random spike generation model also in the encoding scheme. For the decoder purpose, we have the spike or the Purkinje cell output in the form of spikes, which was converted or rate coded into time bins. And this kernel matrix was used or transformed using a summation function as well as an activation function. So here the activation function used was the sigmoidal activation function, and given to a MLP kind of network to come up with the output. So here we use three layers, that is an input layer, a hidden layer, and an output layer for the decoder purpose. And from there, we could reconstruct. So the green color shows the mossy fibers, the red color shows the granule cells, and blue represents the Purkinje cell. So based on the number of granule cells that were number of mossy fibers, the granule cells were designed, and based on the output, the number of Purkinje cells were designed. And for this example, we used a spike generator as a mossy fiber, and that was a random spike generator. And for the granule cell, it was observed that most of the granule cell remained silent. So that was also specified in literature that it is important for learning purpose, or this contribute more for learning. So when you have more quiescent granule cells, it is known to have more learning happening. And the Purkinje cell, based on the granule cell or granule input that comes up, increase the firing rate also. And the learning, so again, coming to the learning part, again, there are different types of learning that is present. Hebbian-based learning in finite precision algorithms, and HTP algorithm, that is high threshold projection algorithm. So these Hebbian-based learning was the simplest, and we could do the same with our data as well as the network. For the finite precision, the spike timings was dependent on the first spike that occurred, and based on which it tends to move or adjust the pre-spikes and the post-spikes. Whereas in the case of HTP, that is high threshold projection algorithms, there the based on the number of iterations, it trains itself to come up to a somewhat a similar kind of behavior as observed in the studies. And so we went forward looking at the firing behavior and the weight change, and it was seen that the mossy fiber as well as the granule cell synapse, so MFGRC synapse, is known or shown to be a slow learner during the training process. And you can see that there have been only a small or the weight change has been very limited. Whereas in the case of the Purkinje cell, you can see there is a PF, an increased learning that is happening, and it is shown with the help of this blue and red lines. And the next step was we needed data, robotic arm data. So we developed a low-cost robotic arm with a simulator in our lab. And this was the interface for that. And we could download it or as Excel file and use that as the um, data set for the further training of the network. And so this is so this is the arm, and it was so it we use these aluminum sheets and motors to come up with a low cost arm, and then that was used to pick up a small cutter or a sharpener, and then place it back at a particular position. So we could do that. And the next step was to identify how much information is being quantified in each of these neurons. So there are two different measures we looked at. One is the sparseness index, and the second one was the storage capacity. So sparseness index will help you identify how much learning happens inside the neuron. So that was the first step. So during and we calculated the sparseness for the granule cell, as granule cell is known to be distributing the sensory signals. So we can see that before the learning process, there was the neurons either responded to all the stimuli or it did not respond to any of the stimuli. Whereas after the learning process, a clear generalization as well as learning interference was seen with the training, and the sparseness index was an average sparseness index was coming around minus or 0.7. So which is also accounted to be a good sparseness index for a neuron. In the case of storage capacity, so the storage capacity definition as such is storage of input-output associations in the synapse. So the Purkinje cell, being a bigger neuron compared to the granule cell, had the option to capture all the information or input-output association in the synapses. And we can see that it was tested and it was finalized. So based on this firing behavior, the storage capacity was calculated around 0.3, so 0.1 to 0.3. So it suggests that around 5 PFPC synapse was required to store at least one input-output association. So that was so a single Purkinje cell actually can hold in a number of input-output associations. So that was the finding which we could get. And in the case of granule cells, we could see that the storage capacity was very limited, and after might be a set of firing itself, the storage capacity reduces very drastically. So we couldn't count storage capacity as a measure for the granule cell, but we tried it for the Purkinje cell. So now the components were ready, and now the next step was to come up with the detailed abstraction model where an encoding scheme, a cerebellum-inspired neural network, as well as a decoding scheme was administered, and from there we could come up with a trajectory plan. So the case study involved with around 21 mossy fibers, 279 granule cells, 6 Purkinje cell, and total adding up to 306 neurons. So this was not just a classification method, it was a prediction for the trajectories also. So the one of the other main objective was we wanted a generalized algorithm which could do multiple tasks, multiple independent tasks, which are of different mathematical formulations. So the second portion was the prediction part. So we had 6 different motors for the, we used a robotic arm with 6 degrees of freedom. So that is why we had 6 Purkinje cells. And the input was 3, that is the XYZ coordinates, that is the end effector coordinates, which conferred to around 21 mossy fibers. And we also looked at the convergence divergence ratio for the MF-GRC as well as the PFPC synapse. Before we went into the algorithm as such, we tried with the robotic arm data on classical machine learning algorithms like naive-Bayes, nonlinear SVM, linear SVM, etc. And we could see that the arm data was of a satisfactory level based on the different efficiencies. And we also tested our algorithm with the same data set as well as different data set. So based on the number of features and number of instances, the time was also increased. And we also created a dummy data set for which is a play tennis data set, which had only 4 features and for 14 instances. And in the training phase, there were 9 instances. Testing phase had only 4. So we used a 66 percentage split. And then we found that the algorithm which we developed had around 72% accuracy. And we also compared this with Cavato model, a multi paired forward inverse model, which was also developed in the lab. And we could that was in the initial phase. So we could move to a 2 data point with a variability of less than 3 centimeters. And for a 4-pointer, it was 7 plus or minus 4 centimeters. And the error was also reducing with the number of iterations. So this is shown only up to 50. And it was easy task at that time. As the number of neurons were increased, the timing also increased. And we can see that still there was only a small difference between the motors. So this portion, the up portion shows the motor error and the down portion shows the sensory errors. So the sensory error is the change from the end effector coordinates, so the actual as well as the predicted end effector coordinates. Whereas in the case of motor error, that was based on the differences in the actual as well as the predicted motor values. And the initial phases still around 40 or 50 iterations, it tend to have differences, and after which it reduces on a like a gradient based. And this was also tested using the kinematic approach. So in the kinematic approach also, we could have the left as well as right positions being taken, as well as we could reach to the desired position. This again, this algorithm can be tried on with the with a gait data as well as for predicting what is the emotional state, or as well as we could come up with where the next move will be. So based on the previous state, one can come up with the desired state. So that kind of a prediction can also be done. Along with that, we have also initial phase of it was done using the EEG data also. So where we have the EEG data for spectral activity data for the relaxed state, left movement as well as a right movement. And based on the data, we could also come up with the left versus right classification also. And beyond all these Mar-Albus Ito theories, now we have a lot of other things coming up. So we have new cells that were identified, like the Lugaro cells, the unipolar breast cells, and so forth. There are recurrent loops that are happening in the cerebellum. So inside as well as to the outside. the plasticity. So earlier, when Mar Albacito had put forward, they were only looking at the PFPC synapse plasticity. But now we have come up to around 16 types of plasticity throughout the cerebellum. So that also has to be added. And recent studies, Wagner 2017, as well as one by Eric in 2020. So in both the cases, they have come up with the reinforcement learning for the granule cell as well as for the climbing fibers. So in generally, it is a supervised learning technique, but in instances, they tend to switch to unsupervised or reinforcement kind of learning techniques. And now there are multiple forms of learning also being identified, which need to be implemented or which need to be. So just like Professor Srini said, we have to explore these options also. So with that, I like to conclude. So